I will share. Just a second. Yeah. Okay. So today we will be discussing about cell culturing. So yesterday we have just got a glimpse of what is tissue culture, what is cell culture, what is organ culture. So all these things in brief we have discussed. So today we will be discussing in detail. So if you don't understand anything, you can just stop me, okay? And you can ask. So cell culturing is nothing but culturing of individual cells rather than a complete tissue or an organ. Okay. So what is cell culture? So cell culture refers to the removal of cells from an animal or plant and their subsequent growth in a favorable artificial environment. So first what we have to do is you have to select a particular tissue from where you want to grow the cell. Suppose you want to grow liver cells. So first you have to isolate a liver tissue. Okay. And you have to, in tissue, as I said, all these cells will be in a tight junction. Okay. They will be like a single layer which will be attached tightly to each other. So first of all, we have to remove the cells into individual cells from the tissue, and then we have to grow them individually in the artificial environment. So we can remove the cells from mouse, mammals, embryonated eggs, okay, at the stage of differentiation. So from any of the sample, okay, this is the specimen from any organism, you can just first isolate a particular organ, okay, which will be called as explant, okay. So from this explant, you are not taking the complete organ, you are taking a particular tissue, some part of the organ, and then that organ can that tissue can be finely chopped either mechanically, that is manually. You will have the equipment using which you can finely chop them. Once you finally, though you chop them, you cannot get the individual cells because the cells are very small in size, which are not visible with our naked eyes, right? So first we have to chop them into small pieces. Then we have to digest them using some enzymes like trypsin, pepsin. So yesterday we have discussed, right, some of the enzymes. So what these enzymes will do, they will digest the uh, what are the attachment between the cells. So that attachment will be lost. So what happens? The two cells will become separate. Like that, the complete tissue, whatever the cells are present in the complete tissue, all the cells will get separated. Now every cell is acting as an individual cell and you can culture them. So once you are getting the individual cells, then you have to grow that cells in a suitable media. So yesterday we have discussed different types of media like DMEM, RPMI 1640 or MEM. Okay, hands. Also, so many media are available, commercially available media are there. So you can choose any, but randomly you should not choose. So each cell culture, each cell has a suitable media, culture media. So which is already established. So much of research has been done on the cell culture. So if you refer to the old papers or old protocols, we can easily know that, okay, why this, which cells are growing in which type of media, and why also we have to know, okay, what is the reason why only these cells are growing in this particular media. Okay, once we know, once we have the knowledge of suitable media, then you have to procure the suitable media. For that, you have to add the media in a flask. So yesterday we have already seen the flasks also. So in that flask, you have to add the media, you have to add the cells, and you have to incubate that at 37 degrees centigrade for 1 day. Okay, first initially 1 day you have to, at 5% carbon dioxide. So you have to, there are some specialized incubators which are designed for cell culturing. Okay, that will maintain the gas also, that is 5% carbon dioxide will be maintained in the incubator. So then after 24 hours, you have to check whether these cells got attached to the bottom of the flask. At the bottom of the flask, you will have a substrate which is already available in the flask. No need to add anything. Okay, once the cells get settled, then they will start growing. Okay, so it will take some 24 to 48 hours for settling. So once it gets settled, then you can see the growth in the cells. They will divide. Okay, one cell become two, two become four, like that. Okay, this is overall how it happens. So this, whatever the cells which are like our tissues, like if you take the skin cells or liver cells, lung cells, or breast cancer cells, or any other cancer cells, so most of them are, they grow in monolayers. So what is monolayer? If you take our skin, so it is like a smooth single layer, right? We don't see any clumps on the skin, right? So that kind of growth is called as a monolayer culture. But whenever these cells becomes cancerous, then they will be starting to grow outside. Then only we will get small, small clumps, warts, like that, right? If we some kind of outgrowth is there on the skin, definitely we will uh, like all are not cancerous, of course, but some change has happened on the within the cell. So that is what we understand. So in the same way, normal cells, they usually grow in monolayers. So only single layer of cells we will see. But some cells, like transformed cells, that means in the within the cancer cells only, the cells which are completely transformed. So here one term is there, that is contact inhibition. That means whenever we do cell culturing, you have a small plate. So in that plate, initially when you add few cells, suppose first day you have added 10 cells. Next day, these 10 cells will become 20. Next day, 20 becomes 40. So like that, they will spread all over the plate. Once the complete plate is filled as a single layer, there will be no outgrowth, because there is a contact inhibition. That means once two cells come in contact with each other, there will be inhibition of the growth, right? So once a single layer is formed, 100% it is covered, then the cell starts dying. If you don't remove the cells, they will die. But this is not the case with the cancer cells, because cancer cells will lost the capacity of contact inhibition. That means even though the cells come in contact, they don't stop their growth. They will be continuously dividing, and you can see small, small outgrowths. Okay, small, small clumps here and there. So that kind of cell growth you can see in the cancer cells. So within our body also, whenever in one part, if you get cancer, so that cells will travel to another part, and they will again, that is called as metastasis. So that is very advanced stage, of course, and not seen in all cancers also. But there is a condition called metastasis, where cells from one region, they will go to the other region, and again they will infect the other region. So that kind of cells are nothing but they grow in suspension. Okay, suspension means they will be floating, they will be moving here and there. And not only the cancer cells, our blood cells. So blood cells, they don't form a layer, right? So in the blood, they will be freely floating here and there. So such kind of cells we have to culture in the suspension. So okay, so this is the overall just basic like idea of how we have to culture. Then we can do subculturing, all these things. Again, we will discuss in detail. So this is first of all the stages of culturing. So first is we have to isolate the tissue. Once you get the tissue, you have to disaggregate. Means you have to make them into small pieces mechanically. Once you do the pieces, then you have to add enzymes. Then you will get a single cell. Okay, like many number of single cells which are not attached to each other. Now that stage is called as primary cell culture. Okay, so primary cell culture always have a limited lifespan. So you cannot culture them as long as you want. So it will have a particular lifespan. So after 10 generation or 20 generations, that will die. Okay, so limited lifespan, and they will undergo senescence, means death. Okay, so finally they will undergo death after some period of time. So once the primary culture has happened, then they will convert into finite cell cultures. So once you culture them over many times, so there will be some changes and they will become finite cell cultures. Then finally they will undergo transformation. So transformation is not, they are called transformed cells. Means some changes in the cells takes place and they will become continuous cell lines. Continuous cell lines means they are immortalized. You can culture them for any number of times. They will become, especially cancer cells. Okay, they undergo transformation. So yesterday, if you remember, I told you about HeLa cells. So a woman called Henrietta Lacks. So she was diagnosed with the cervical cancer in 1952. So at that particular time, the doctor isolated the tissue, cervical cancer tissue, and he disaggregated the cells, and he made into primary culture, then finite culture, and finally it became a continuous cell line, so that from 1952 till today, see how many years has passed. Okay, so till today still we will get HeLa cells and we can culture any number of times. Okay, and still that may continue for any more much more years to go, right? So they become continuous cell lines. So this is the definition. You have to remember what are primary cell cultures, finite cell cultures, and continuous cell cultures. So this is how the growth curve of the cell culture. So same thing, whatever I have explained, this is in a graphical manner. So weeks in the culture. So x-axis representing the weeks. So 2 weeks, 4 weeks, 6 weeks, 8 weeks, like that. So in the primary culture, initially the cells, they are slowly, they are adjusting because from our body, they are going into the lab. So there will be definitely changes in the environment. So they have to get adjusted. So it will take some time to adjust and then they will start growing. Once they become attached to the flask, they will grow. Then we have to do subculturing. Subculturing means again we have to remove them from the flask. Again we have to divide them into two flasks. So that is called subculturing. After subculturing, again we have to do second subculturing. That means from 2 flasks we have to remove and we have to add them in the 4 flasks. So like that, once we do, after second or third week, after second culture, so the growth will be like exponential. It will be very fast. So see, you have to culture these lines, they are subculture intervals. Okay, so slowly we have to check for the growth of the cells and we have to subculture continuously. Finally, after 12 weeks, around 13th week, they will become transformed cells. They will undergo transformation. So till that time, that cells are called cell lines or finite cell lines. Once they undergo transformation, they will become continuous cell lines. Excuse me. They will undergo continuous cell lines. They can grow any number of times. But if we are not properly maintaining all the conditions or any changes, sometimes they also may undergo senescence and death, and they will drop down. Their growth won't, or any contamination. So all this can happen. Or they can, if everything is properly maintained, it can become continuous cell lines. So this is how the culturing takes place. So what are the different disintegration methods? So that we will see. So once when the tissue was isolated, it is dissected. Okay. We have to remove the fat cells, necrotic cells, all these things from the tissue. and we have to get only the cells of our interest. Only that particular tissue has to be taken, and you can disintegrate by using three methods. First is fine dissection. So you will have small scalpel like of things, small knives. So by using which we can chop them into small pieces. Then now that is called as the primary explant. Okay. And that gives you the explant which can be directly transferred, and you can go for the secondary explant culture like that. Or if more amount of growth is there in the tissue, you can go for the subculturing. This is just dissection. Then second is mechanical disaggregation. So in the mechanical disaggregation, if you do pipetting, vigorous pipetting, so keep on very quickly if you do, then also there will be outgrowth. Okay, like if you vigorously pipette the cells, the tissue, then the cells will get separated because of the friction. Okay, or you can do with the syringe also, or sieving. You have to take a filter. There will be filters available. So on that you have to keep the tissue piece and you have to just sieve that. So slowly small pieces will come. Then that whatever the small pieces you are getting, they are called dispersed primary cultures, and they will undergo subculturing and you will get the cell lines. This is mechanical disaggregation. Then the third one is enzymatic disaggregation. So in the enzymatic, you are using the enzymes. So you can use trypsin or collagenase. So you have cold trypsin, warm trypsin, and collagenase methods. So cold trypsin means you are taking the trypsin in the cold condition and you have to keep overnight incubation. You have to add some amount of trypsin, cold trypsin. You have to add to the cells. You have to keep it for incubation, overnight incubation. Then you will get the primary cell cultures, or you can go for warm trypsin. Okay. So warm means you have to increase the temperature slow little bit for the room temperature, then you have to use them. Then collagenase. So collagenase also is another enzyme, so which will break the uh, what you call the attachment between the two cells. So once you add these enzymes and you keep it for the proper incubation, then you have to centrifuge, you will get the pellet. So that pellet we can suspend in media and you can go for the subculturing and we'll get the cell lines. So first isolation of tissue. First how to isolate the tissue. First we have to sterilize the site. Suppose you are taking a tissue from the mouse. First you have to clean the site with 70% alcohol. Always you have to maintain aseptic conditions because there is a chance of contamination. Okay, so first you have to rub that site with the 70% alcohol and slowly you have to cut like surgeries how we do, and we have to remove the tissue aseptically. Then you have to add a media called transport media that we have to transport to the lab. If you are delaying, suppose the animals are there in some other place and your lab is there very far, so within 24 hours, suppose if you cannot do, like if you can, or there is a holiday in the next day or something like that, usually in the research we can expect some kind of delays. So if there is any delay, you can store the tissue in 4 degrees centigrade for up to 72 hours, that too in transport media. Then you can transfer to the lab. So this is how the chick embryo is isolated. See how they with the using the forceps, slowly they have isolated the embryo. Slowly they have, first they are rubbing with the 70% alcohol, then breaking that, then slowly by using the forceps they are removing the chick embryo. This is the process how they will isolate the tissue. Then you have to go for the, once you got the tissue, suppose here you got the embryo, okay, Then you have to disintegrate. First is disaggregation, enzymatic disaggregation. So you can use warm trypsin, that is at 37 degrees centigrade for 30 minutes. We have to incubate. If you keep it for more long periods, then the cells will die. You should not, because that will damage the individual cells also. It's an enzyme, right? So you have to stop the damage. So what you have to do immediately, you have to add the media, then it will be neutralized. Okay. Or other thing is you can use cold pre-exposed, means already you have to cool the trypsin in the fridge, and then you can soak that cells in this cold trypsin for 4 degrees centigrade for overnight, and then you can transfer them to 37 degrees centigrade for less than 30 minutes. Like that also you can do. But here advantage is you will get the higher yield of viable cells. So the cell damage will be less, and you can preserve many different types of cells. And other enzyme other than trypsin, trypsin is very commonly used. Okay. Other than trypsin, you have collagenase. For connective tissues, muscle tissues, fibrous tissues, you can use collagenase. Other than that, you have pronase, dispase, DNase, hyaluronidase. So that's what I'm saying, like based on the tissue you are using, you have to select the enzyme. It is not that blindly you should go and add trypsin. It is in your hand that you can choose any of the enzyme based on your experiment. Then you can also go, if because if we use enzymes, definitely there will be a chance of damage to the cells. So then you can also go for the mechanical disaggregation. So this is by scrapping. You will have a scrapping equipment. Equipment means a small plastic scrappers. So by using that also you can just scrap. You can do spillage or sieving using filters or syringes, pipetting. So any of that you can use. So this is how the scrapping. You will have the scrappers by which you can just scrap the cells. This is sieving. By using a filter, we can sieve. This is syringe. This is pipetting, right? Then culture conditions. Okay, you got the cells from the tissue. Now how to culture? So what type of conditions you have to maintain? So this also, it varies from cell type to cell type. So first you have to select the media. Which type of media is suitable for this type of cells? So that media should contain amino acids, carbohydrates, vitamins, minerals, growth factors, hormones, then gas proportion like 5% carbon dioxide and 95% oxygen. That has to be maintained. And pH, osmotic pressure, temperature, all this should be maintained. So the best example is in summer season, definitely the temperatures will be more. Even in the labs also, the temperature will increase. So properly we have to maintain 37 degrees centigrade. And even suppose if you want to create a hypoxic condition, that means no oxygen or very low oxygen, then what you have to do, you should not keep 95% of oxygen. You have to reduce the percentage of oxygen so that you will get that particular environment. So like that, you have to alter the environment based on the external environment as well as your experiment. So this is the media. Okay. So the culture media are like common basal medium, which includes Eagle minimal effective medium, Dalbecos modified Eagle medium, RPMI 1640, HAM F10. So yesterday we have discussed all these medium are available. All these media contains amino acids, glucose, salts, vitamins, minerals, all are present. But L-glutamate, it is not present in all. So if you want, you can add. Then serum also you'll get separately. If you want, you can add, or if you don't want, you no need to add. Even antibiotics, fungicides, everything you will get separately. So that we have to add as per our requirement. So once we add them, then it will become the complete media. Okay. So Serum may or may not be used. It's our wish. If we are using, so frequently we use fetal calf serum, fetal bovine serum, okay, or sometimes horse serum also we can use. So why we need to use serum? So yesterday I told you, because serum contains all kinds of nutrients for which will promote the growth of the cells. So, but you cannot define each and everything. You don't know exactly what is there in that. Means you cannot define everything. This is 1 gram, this is 2 gram. Like that, you cannot say. We have everything, but we don't know the quantities. So it is sometimes advantages, sometimes disadvantages. So for just for growth purpose, it is very, very good. You can add the serum. And then antibiotics and fungicides. So definitely when you are culturing, maybe whatever, how much ever care we take, there is definitely a chance of contamination. So to prevent the contamination, we use antibiotics, fungicides. Fungal contamination is also very common, and antimycotics also we use. So there is mycoplasmal contamination is a very serious problem in cell culturing, because the mycoplasma, it is similar to the eukaryotic cells, and we don't know, we cannot even under the microscope we cannot differentiate whether it is the animal cell or mycoplasma. So slowly it will spread. So that is a very big problem. So usually we also include antimycotic agents. Okay. Then antibiotics, the bacterial or fungal contamination we can easily detect under the microscope, but still we use these antibiotics and fungicides in the media. And some types of primary cell cultures, they require other supplements like collagen, fibronectin, epidermal growth factor. Means because it has to get attached to the flask and then it has to grow. So for that attachment purpose, it requires some additional requirements. So if required, we can add. And then media, first thing is what we have to do is we have to sterilize everything. Okay, that is the first step, initial step. And different sterilization techniques are there, like autoclaving, filtration, all these things. So usually in the laminar airflow, we use ultraviolet rays. First we wipe it with the alcohol, the surface, and then we on UV rays. So after 15-20 minutes, we can just off the UV light and we can again wipe it with the alcohol, and that gets sterilized. Then all these, whatever the flasks, then pipe, tips, they will be already sterilized. In the packets we will get commercially. So no need to sterilize again. If you want to sterilize, you have to keep them in the boxes and we can autoclave them. Then the heat labile material like media, all these, you just cannot heat them in the autoclave. So you have to filter them. So first you have to filter all the media or antibiotics or whatever is required to sterilize. So all that you have to filter. Only after filtering you have to prepare the complete media. Even after filtering or everything, it's always better to keep only media without adding any cells. Just to keep the media in the flask and keep it for 24 hours or 48 hours at 37 degrees centigrade. Suppose by any chance it got contaminated, if the media is contaminated, you can see the bacterial or fungal growth in the media before 48 hours. If there is no growth, okay, if it is clear, perfect, then only you can go for the cell culturing. Because if the media is contaminated, the entire cells, everything will be lost, because it will be spread completely to all the cell cultures, and from cross contamination also can happen, right? So these are the important techniques which we have to follow in the cell culture. So these are the culture vessels. So these are the flasks, this type of flask, and you have different sizes. This is called T25, T75, T100, like that. T25 is small one, T75 is much bigger, a little bigger than T25. Like that, you will have different sizes. So based on how many number of cells you want, you have to do. Suppose in T25, you will get some 4 to 5 lakhs of cells in T25. So if you want 3 times more, you can go for the T75. Like that, you have to choose. This is for the suspension cultures. The other one, round bottom one. So in that, you can go for the suspension. So modes of propagation, that's what, like we have adherent cells and suspension cells. So depending upon their origin, the animal cells grow either as adherent monolayers or suspension. So I am taking only one example. Suppose you want to culture the skin cells. They are adherent. They always attach to one another and they will form a layer. And if you want to culture the blood cells, of course you cannot culture in the sense because they cannot propagate. It won't contain nuclei, so they cannot propagate. But for example, I'm telling blood cells, which will be in suspension. So you can go for the suspension culture. So adherent cells are anchorage dependent cells which propagate as a monolayers, which are attached to the cell culture vessels. Okay, so usually cells which are derived from the tissues that we can culture as a adherent cells. Then suspension cells, they proliferate without being attached to the substratum. So that you can, so in the other type of flask you can do. Example, hematopoietic cells or cells derived from the tumors or cancers. So this is the primary culture. So how they look, see how they are attached to each other. So they are capable of only limited number of cell divisions, primary culture. When they are placed in the culture, so directly from the tissue, then they are called as primary culture. Okay, immediately after isolating them from the tissue and you are placing in the culture, then they are called as primary cultures. So until you subculture, first subculture, they are called as primary cultures. So these are the epithelial cells. Examples, you can see very beautifully colorful. So you can see all the cells which are tightly attached to each other. So you have primary cell cultures are of many types. So like mouse embryos, chick embryos, human biopsy material, then transplantable animal tumors, chick embryo, organ rudiments. So from chick embryo, if you take the heart, liver, lungs, kidney, brain, spinal cord, skin, so everything you can go for the primary cell culture. Then once the primary cell culture are established, then you can go for the subculture. See here, you can see how the person is handling. Okay, so usually the flask will be like this, horizontally placed. And here you can see at the bottom where the tip is attached. Okay, that bottom layer will have the substrate and there the cells will be attached. And above that, usually the media will be floating. If you want to remove, you can just use this pipette. You can just you add trypsin or collagen or whatever, collagenase or whatever, and you have to remove that, and then using the pipette along with the media, the cells will come out. Okay, so then you will go for a subculturing. Subculturing or splitting cells is required to periodically provide fresh nutrients and growing space for continuously growing cell lines. Right? The frequency of subculture and the split ratio or density of cells plated depend on the characteristic of each cell line being carried. So not all cells, it is not that after 24 hours you have to do or 48 hours you have to do. It is not like that. You have to, every day you have to check. Okay, that means as I told you, first day, today, suppose I have removed the cells from the tissue. I have mixed that with the media and I have added in the flask. So today I won't do anything, just I will incubate. So today and maybe tomorrow also, I will check only whether that cells got settled or they got attached or not. Once today, tomorrow also is finished, then day after tomorrow, then the cells will get attached and they will start dividing. So first day, today I added 10 cells. So that 10 cells won't become 20 tomorrow. First they will get settled. Then only after 2-3 days, they will become 10 will become 20, 20 will become 40, 40 will become 80. And finally, after maybe 4 days, the entire flask will be filled with the cells. So suppose if it is almost 80, that the term we use is called confluency. That means confluency means the area of the flask, how much it is covered by the cells. That can be, suppose first day it is 10% confluent. After 3-4 days, it will become 50% confluent. After 6 days, it will become 80 or 100% confluence. So before it reaches the 100% confluency, I have to remove the cells. If not, what happens? And again, just remember, once the cells get attached to the substrate, we have to change the media. Every 2 days or 1 day, we have to change the media, remove the old media and add the fresh media, or else the cells will die. So once the cells reach 80-90% confluence, I have to subculture. If not, what happens? If once it reaches the 100%, there is a contact inhibition. So the cells stop dividing and they will start dying, and cells will float in the media. Okay, so we should not go till that point of time. We have to remove the spent media, add fresh media regularly, and once it reaches 80% confluence, you have to remove the cells and you have to subculture.
Ma'am, is
So,
there any
yeah.
difference between confluency and colony formation?
Yeah, colony formation is different. That is essay. Usually that is a particular experiment. Colony formation is same. That is like colonies. Each cell will form a colony. Okay, like in microbiology, we see each cell, it will divide and forms into small colonies. But here we use the term confluency. That means how much percentage of the area of the flask is filled with the cells. Here we don't see any colonies, right? Is it clear? Here we see a continuous layer,
Yes,
no
ma'am.
question of colonies. We will do colony formation essay in this cell culture also, but that we will do in the agar plate, same as the microbiology experiment, how we do. So like that we will do, but that we cannot use here. The name here we term confluency, right? So both are different,
Okay,
right?
thank you.
So here subculturing is different for adherent cells and suspension cultures. So this is how we do. See here, okay. So cells, here's subculturing of monolayer cells. So first, what you have to do is, after 1 week, you got a layer of the cell, right? Single layer like this it will form. You can see single layer of the cells. Then you have to change the media first, and you can wash them with the PBS, phosphate buffer saline. Yesterday I told you phosphate buffer saline while we were discussing. It is a basal media. Then you have to add the trypsin, incubate for some 10 minutes at 37 degrees centigrade. Now you see all the cells are separated. Now once the cells are separated, immediately you should add the media, because the media will neutralize the trypsin. So there is no more separation. Already they are separated. Now see, always the flask should be placed like this, horizontal, and the cells get attached at this position, at the bottom layer of the flask. Once you add the media, you should keep it in the vertical position. Okay. And here again, you have to remove everything, all the contents in the tube. You have to centrifuge. You will get the pellet. And to the pellet, you should add the fresh media, and that you can split into two flasks. So that is how we do subculturing. So is it clear, or should I explain again?
Clear, ma'am.
Clear now? So from one flask, You are removing in one flask, you got 100 cells. After you add trypsin, all 100 cells are removed, separated. Then you add media, centrifuge, you are getting pellet. To the pellet again, you are adding the media. So now in the media, you have 100 cells. Now you are taking two flasks instead of one flask. You are adding 50 cells in one flask, 50 cells in other flask. So now how many each cell, each flask you will get 100 cells again after 3-4 days, right? So this is subculturing. Again after 3-4 days, from each flask you will separate. Now 100 cells again split it into two flasks. Now you are getting 4 flasks, each with the 50 cells. So this is called a subculture, right? Then
Yes.
continuous cell lines. So after first subculturing, the primary culture is called as secondary culture. Okay. And thereafter it will become continuous. And it will become the continuous cell lines. So then they will become immortalized. That means there is no death until you keep everything perfect without any contamination, proper conditions are maintained. Easily you can proliferate the cells for any number of times. And they will undergo some random mutations and modifications, and also they will express telomerase genes, and they will become immortal. So how our cells are mortal, that means how see normally our cells have they have limited time period. So after some time the cells will die and new cells will replace them, right? We have the mechanism. If not, what happens? We might have in a very big size, because if the growth of the cells are not stopped, so what happens? We don't have this unlimited or what you call indefinitely we might have grown into big size. It's not that. So the cells will stop after certain period of time, and again new cells will form, like blood cells. After 120 days the old cells will go and new cells will come, right? Even the skin cells, every 24 hours they will the old cells which are damaged, everything they will go and new cells will come. But that is not there here. So they will be continuously growing. So that kind of cells are called continuous cell lines. So here the established cell lines are, they can be normal also. So don't think that transformed cells means always they are cancerous. No, they can be normal cells also, but they have become transformed because of some mutations, but they are not cancerous, right? Or they can be tumor cells also. So here are the examples. HeLa, I told you, HeLa cells. So they are from human Henrietta Lacks. So it is cervical cancer cell. So 293T, it is from kidney, embryonic kidney of human. Then A549, which are lung carcinoma cells. Then ALC from bone marrow of murine. Then CHO, Chinese hamster monkey, okay, ovary cells. Then HP54, which are hybridoma cells. FM3, they are metastatic lymph node cells. So these are already well established and they are available. If you want to perform any, suppose I have got a drug, new drug, and I want to test it whether this drug is acting on the cervical cancer. Directly I can procure this HeLa cells. I can keep order and I'll get the HeLa cells. You have the labs which can provide, and I can culture the HeLa cells and I can use the drug for the treatment and I can get the results. So like that, all these cells are commercially available. If you want to perform an experiment, we can get them. So this is how, see, monkey kidney cells, sorry, mouse kidney cells. Then you have secondary hamster kidney cells. So after splitting for several times, you have got that morphology also will changes sometimes. Then incubation condition. So cell cultures are incubated at specialized incubators which will regulate the temperature, because they will have the water jacketed incubator. That is, we have to give the water. We'll get specialized trays in which we have to fill the water and we have to keep. And we'll have the cylinders, carbon dioxide, cylinders, so which we have to supply to the incubator, and the 5% carbon dioxide is maintained, 37 degrees centigrade is maintained, and proper humidity also will be maintained in the incubator. Then, as I told you, sterilization is very important. So how the medium is sterilized? So various media constituents and reagents, they have to be sterilized very carefully. So which are very resistant to heat, like metals or petri plates, glass, that we can do it for autoclaving. And media or proteins, trypsin, some antibiotics, such kind of things, you can go for the filtration. Heat stable, like water, salts, supplements, like tryptase, not trypsin, okay, tryptase, peptone, such kind can be autoclaved at 121 degrees centigrade for 20 minutes. But heat labile constituents like serum, trypsin, proteins, growth factors, they have to be sterilized by filtration through 0.2 millimeter porosity membrane. We'll have the membrane filters. And each filtrate should be tested for sterility. I told you, like, we have to check for any contamination for 24 to 48 hours to avoid failure due to contamination. Okay. Then cryopreservation. Okay, I have done all the, like, I have cultured the cells, but unfortunately I couldn't perform the experiment because somewhere I am going somewhere or something. I want the cells to be used after some more time, or I'm transporting the cells to some other person, or so, like that. So in such conditions, what we have to do, we should not like discard, or we should not do that. We can store the cells, where, like, by using a process called cryopreservation, that is freeze preservation of animal cells in the cell line banks. There are cell line banks, and every lab also, usually big labs, will have the cryopreservation. Liquid nitrogen facility will be there. So a cryoprotective agent like DMSO or glycerol should be added to minimize the injury to the cells. So there is two techniques, freezing and thawing. Freezing means keeping in a very cold condition. Thawing means again getting back to the room temperature. So that if you do it very frequently, the cells will die because they cannot adjust to both the temperatures. So even freezing also, you cannot directly, like, from room temperature, you cannot keep them in minus 196 degrees. First you have to keep that in 4 degrees, then minus 20, then minus 80, like that. You have to slowly, one day you have to keep in 4 degrees, next day minus 20, then next day minus 180, like that, minus 80, then minus 196, like that. Slowly have to transfer, and again getting back also in the same way. First you have to keep in minus 80, then minus 20, then 4, then room temperature, right? So to prevent, during this process also some cells will die, all cannot get adjusted. So to prevent maximum prevention, we can use DMSO, dimethyl sulfoxide, or glycerol. And we first, in the small, you will get 1 ml tube, you have to add the cells with media, and above that you have to add the DMSO. It won't mix with the media, it will form a layer above the media, and that you have to seal it with the paraffin. And that ampules, you will have small cans, like liquid nitrogen will be there, and in that you will have small pockets where you can keep these small tubes, and you have to store that at minus 196 degrees centigrade. Okay, so you can store them for very long years together in that. This is called cryopreservation. So again, after, once we get once we freeze them, again you have to thaw them. Thaw them means getting back into the culture again. So how you have to do? First, check cell line data sheet. First you have to look for the data sheet. Suppose, see, I have stored, so after 100 years I may not be available to tell everything about that cell lines. Some other person will be taking care of that cell. So I should maintain a data sheet so that any person, even after 50 years or 100 years, if you read the data sheet, that person should understand everything about the cell lines. What are the culture conditions? So what are the cells? We have to label everything. When I have stored that, all these things we have to label. And accordingly, we have to prepare the flask. We should know which media is suitable, all these things. Then first you have to pre-warm the media. Then you have to collect the cells. You have to allow to thaw. That's what I told. Like from minus 196, directly don't get it into the room temperature. So slowly after 2-3 days, it has to come to the room temperature. Then you have to pre-warm this media. You have to dilute it and add that to the cells should be added to this media, and we have to examine the cells under appropriate temperature. We don't know whether simply you cannot just directly add the cells and wait for them to grow, because we don't know whether the cells are alive or dead also. We have to first examine the cells for 24 hours, then only we should plan for the experiments. Okay, directly don't plan for the experiments and then you have to thaw the cells. No, first you have to check the cells. And also we should look for the contamination. So contamination is nothing but the growth of the any of the microorganism or any unwanted organism which can be there. So yeast, fungal or viral growth or bacterial growth or mycoplasma, I told you. So all these, any growth can be possible. And sometimes we'll have the cross contamination also. So in big labs, usually, like if the same person is handling 2 to 3 types of cell lines, or if within the same cabinet, if 2 or 3 people are doing the experiment, means not at a time, but one after the other like that. So if properly they are not sterilized, okay, what happens, there's a chance of cross contamination. That means the liver cells may get mixed with the cervical cancer cells, or kidney cells may mix with the liver cells. Like that, the contamination of one type of cell lines with the other type of cell lines is also possible. That is called as cross contamination. See this, by looking at the media flask also, you can easily identify if there is contamination. First thing is pH. So in that, I told you, whenever you have seen, I think if you see the flask, culture flask, the media is in pink color. So I will just show you the slide. Yeah, can you observe here, in the flask the media is in pink color. So why it is pink in color? This is because we use indicators. We have no like pH indicators in, usually in chemistry we use the indicators. So in the same way here also, the pH, to check the pH change, we use indicators. If there is any growth of bacteria or yeast or fungi, the pH will change. Okay, in the bacterial growth the pH will drop. The yeast, the pH will change, it can drop or increase, because it will release the waste, all thing. Then fungi also will change the pH, so the pink color will become yellow. Okay, by looking at the color change you can see. And the cloudy medium, the media you in the flask, you see it is very clear, but if there is any growth of bacteria, yeast or fungi, it will become cloudy. Okay, in bacteria you can see rods, cocci, everything you can observe under microscope. And in yeast they will form round ovoid particles, with small particles you can see black spots we will see. Then in fungi, filamentous mycelium. Okay, spores also usually we see in the fungi. Then cross contamination. So to prevent the cross contamination, always first only one cell line should be added at a time. It should be handled at a time. and separate media bottles or separate flasks like that you have to maintain for each and every cell line, and you have to clean the bench every time. Okay, and how to detect is, you can see morphology. So yesterday we have discussed, and today also the cell, like the different tissues have different morphology, different shape, right? So easily you can detect if one type of cell is mixed with the other type of cell. You can see different morphologies, and the growth rates are different. Okay, or not all tissues grow in the same rate. So one type of cells will grow in at one rate. Okay, and the results you won't get properly. And you have some detection techniques also, like isoenzyme pattern analysis we can do. We can go for DNA sequencing or southern blotting analysis. So like that you can apply different techniques. Okay, sorry, just a second. Yeah, so what are the applications? So applications are, we can use them as a model system. So before performing any experiment, if you want to develop a vaccine or drug or anything, so first you have to perform that in the cell culture. So cell cultures will provide a good model system for studying basic cell biology. If you are not doing anything like any testing or like that, but simple, if you want to study what type of cell it is or what is the mechanisms, basic cell biology can be studied. Basic biochemistry can be studied. What are the metabolism which is going on? All this you can. And interaction between the disease causing agent and cells. Suppose if some type of cell infects, like COVID, you can say best example, how this COVID is infecting the lung cells or heart cells. Like that, you can just check the mechanism, or you can use the drug, how this drug is acting on this particular cell. Or you can go for aging, to study aging, how the aging process, how the cells are getting aged, what are the mechanisms. Or nutritional studies, if you provide iron, minerals, vitamin, like that, you can just check what is the function and how what is happening. So all this you can study by using cell cultures. So other than like drug screening, development and toxicity testing, cancer research, virology, monoclonal antibodies, vaccines, then genetic counseling, gene therapies, all these can be performed in cell culture experiments. Okay, so this is about the cell culture. So what are cell cultures and what are the protocols? So now we will discuss about cell culture media composition. Okay, just a second. I will stop the screen.
మొన్న మేత పాప లేట్స్ పెట్టి మేమే తిరిగినాం. తానియా ఫిష్ ఫ్రై చేద్దాం దా.
Okay, so now we will discuss about the cell culture media. So what are the, please mute yourself everyone.
ఇన్నే కూర్చునుడే ఓలో వేసుకోవాలి.
Please mute your microphone. Okay, so what are the components of the cell culture media? So cell culture media consists of amino acids, then vitamins, inorganic salts, glucose, serum. Okay, all these are the basic general components. So in this we have natural media and artificial media. So natural media is nothing but, in olden days I told you, even now also if you want we can use. So the natural media consists of biological fluids like plasma, serum, lymph, human placental cord serum or amniotic fluid, directly we can use these fluids as a media. Or tissue extracts, like we can use the liver extract, spleen, tumors, leukocytes, bone marrow, or bovine embryo or chick embryo, that you can use. Then clots, plasma clots. So yesterday, whatever the experiments we have just studied, so all of them, they used plasma clots. So even that you can use them as a natural medium. And artificial medium is artificially prepared. We will get the powders mostly, which we can mix it and we can prepare the media, like balanced salt solutions, PBS, phosphate buffer saline, DPBS, HBSS, EBSS. So all these are the Eagle's balanced salt solution. Like that we can get them commercially. Then basal media, MEM, DMEM, and complex media, that is RPMI 1640 and IMDM. So all these are the different media which are available. So what is natural media? Natural media consists of all naturally occurring biological fluids. Okay. So it is very useful and convenient for the animal cell culture because they are natural things. So the cells also will easily get adjusted in the media. But there is a disadvantage because we don't know the exact composition of the natural media. As I told you for serum, you don't know like how much amino acids are there, what is the quantity, or how much carbohydrates are there. We don't know exactly what is the composition. So therefore the reproducibility can be affected. Like you can take embryo extract. So it is a crude homogenate of 10-day chick embryo. So what they have done, they have isolated the chick embryo 10 days old, and they have to mash it. It is homogenate is prepared and then centrifuged. This crude extract can be fractioned into low molecular weight fraction, and this can be directly used as a media. So here they promote cell proliferation and they contain peptide growth factors. And we have another fraction which is high molecular weight fraction, which will promote pigment and cartilage cell differentiation. So it should be always frozen and thawed at least twice. Okay. Then serum. So then we will see the individual components. So in the serum, it contains growth factors, minerals, lipids, hormones, proteins, many things will be present. So usually bovine calf or fetal bovine or human serum also will be used. So sometimes some cell lines, they demand their own serum. Suppose that means they don't grow in any of the serum, but they will grow in only the serum of that particular organism. Like for example, if you are separating it, if you are taking the cell lines from the human, so we have to supply the serum of the human itself. Then only the cells will grow. So some type of cells will be like that. So if the cells are not like that, you can use calf serum or fetal bovine serum. Then we have proteins. So proteins are required as carriers for minerals, amino acids, then hormones. Like here you can see fatty acids. Then albumin is a carrier for lipid. Then minerals and globulins. And fibronectin, which promotes the cell attachment. So all these proteins are very important. Then alpha-2 microglobulin, it inhibits trypsin. Then fetulin is a fetal serum which enhances the cell attachment. Then transferrin, this binds to the iron and make it less toxic and more bioavailable. Okay. And they also increase the viscosity of the medium so that it will reduce the stress while you pipette or stirring. So usually some kind of agitation will be, some kind of stress will be generated. So to prevent that, usually the protein will make the medium more viscous. Then growth factors. So we have natural clot serum, which stimulates cell proliferation. than serum. Okay, and we can also remove them physically. They increase the stimulation of the cells. Then you have PDGF, platelet derived growth factor. So this is from the platelets. During clotting, we get this growth factor. So it is one of the family of polypeptide with mitogenic activity, and it's a major growth factor in the serum. Okay, so it stimulates the growth of fibroblasts and glia. Then apart from that platelet derived growth factor, we also have fibroblast growth factors, epidermal growth factors, endothelial growth factors like VEGF, that is vascular endothelial growth factor, which is involved in angiogenesis, that is blood vessel formation. Then apart from these growth factors, we also should add hormones. Then insulin promotes the uptake of glucose and amino acids and affect the mitogenic activity. Okay, so like insulin like growth factor 1 is there, and insulin like growth factor 2, they will go and bind to the insulin receptor and they will perform in the same way. And this IGF-2 stimulate the uptake of the growth hormone, which is present in the serum, fetal serum. So like this, all these hormones, they have the mitogenic effect. So this is the list, and what are the concentrations which we can use? Proteins, polypeptides, growth factors.
The settings are
Please mute yourself. Yeah, amino acids, lipids, carbohydrates, polyamines, urea, hormones, vitamins. So all this in what concentration you have to use. So all this there in this table. So then I told you in the serum, so we have advantages as well as disadvantages. So what are the advantages of serum in media? Because serum contains various growth factors and hormones. So that will stimulate the growth and functions of the cells. And also it will help in attachment of the cells. Okay, and they act as a spreading factor to spread in the flask. So for that also this serum is used. And it is a buffering agent to maintain the pH, and it's a binding protein, and also it minimizes the mechanical damage which is caused by the viscosity. So these are all the advantages of using serum in the medium. Whereas the disadvantages of serum is lack of uniformity in the composition of the serum. So we cannot define each and every component of the serum. So that is the main disadvantage. And the uniformity is nothing but the composition will change. So the environmental conditions, because the serum has to be collected from fetal bovine or fetal calf. So all the factors from where this calf or bovine is present, all these factors will affect the serum, quality of the serum, like what it is eating. So then what are the environmental factors? Like that, from batch to batch, the serum quality will change. And testing needs to be done to maintain the quality of each batch. And we have in the handlers, the people who are handling the animals, okay, they can also spread the contamination. So we have to look for that also. And they may contain some growth inhibiting factors. The risk of contamination is more because I told you the handling errors. And the presence of serum in media may interfere with the purification and isolation of cell culture products. So these are some of the disadvantages of serum in the media. Then artificial media, so which is commercially available. So artificial or synthetic media are prepared by adding nutrients, organic and inorganic nutrients, then vitamins, salts, oxygen, carbon dioxide, gas phases, then serum, proteins, carbohydrates, cofactors, everything is added in a proportion. And we have different artificial media that is devised to serve one or more of the following purposes. So what are the purposes of using? First is immediate survival. So we can use balanced salt solution with a pH, specific pH and osmotic pressure. If you want prolonged survival, we can use the balanced salt solution, which is supplemented by organic compounds and serum. Then indefinite growth, suppose it's continuous cell lines. So we have to choose the separate artificial media. If you want to look for specialized functions, so then you have a different media. So like that, you have to select the media. And chemically defined media. Chemically defined media is nothing but they contain contamination-free, ultra-pure inorganic and organic ingredients, and also may contain pure protein additives like growth factors. And they are the constituents which are present in the media are produced in bacteria or yeast by genetic engineering technique to provide vitamins, cholesterol, amino acids, and fatty acids. Then conditioned media. So conditioning media adds undefined components and should be eliminated if possible after the active constituents are determined. So it is only for a particular period of time. Okay. So here the scientists, they have showed that the conditioning of culture media that was necessary for the growth and differentiation of myoblasts, so muscle cells, was due to collagen, which is released by the feeder cells. So I told you feeder cells. Feeder cells are nothing but, so in the flask, we have told that like cells are attached at the bottom surface. Sometimes the cells cannot grow. So therefore, first we will use one type of cells called feeder cells, which form a single layer, and above that we culture the cells. So these feeder cells will provide collagen for the growth of myoblasts. So it is supporting the growth of the myoblasts. So using feeder layers and conditioning the media with embryonic fibroblasts or other cell lines, they are very good for culturing difficult cells. So some cells will be very stringent, very determined, which cannot grow in any condition. So for culturing such kind of cells, we can use this conditioned media or feeder layers. Then protein-free media. So protein-free media do not contain any protein. Okay, only non-protein constituents are present. So this will promote superior cell growth and protein expression than serum containing media, and they facilitate downstream purification of the expressed product. So I told you in the first class in the introduction to biotechnology, so we have to express a particular protein of interest and then we have to purify that. First we have to isolate that is called downstream process, and then we have to purify. For such kind of experiments, we usually use protein-free media. The formulations include MEM, RPMI 1640, which are protein-free, and protein supplements can be added if you require. So what are the basic components of complete media? So these components, they vary among the cell lines. So based on the cell lines, we have to formulate. But the components of specific functions are usually present. That is, first is buffering system. So regulation of pH is very important for the culturing of cells. So therefore, pH has to be maintained. The buffers should be maintained. So there are two buffering systems are there. So natural buffering system, like when we are providing the carbon dioxide, it balances with the bicarbonate. So carbon the bicarbonate, carbon dioxide bicarbonate buffering system. So that is a natural one. So the cultures with a natural buffering system needs to be maintained in the air atmosphere with 5 to 10% of carbon dioxide. So usually in cell culture, we maintain 5% carbon dioxide. It is low cost and non-toxic. Whereas we can also use HEPA's buffer. So here a chemical buffering using a Zwitter ion, which is called HEPA's. It's a superior buffering capacity. So it has a pH range of maintenance is 7.2 to 7.4. So here no need to maintain 5% carbon dioxide. So though you maintain or not, the pH will be maintained. And it is relatively expensive and toxic at higher concentration in some cell lines. And it greatly increases the sensitivity of the media and phototoxic sensitivity of the media to phototoxic effects. Okay, whenever you have fluorescence light is used, so there will be phototoxicity. So this HEPAs will increase the sensitivity. So therefore, it can be toxic to some of the cell lines. Then what amino acids we have to use? So essential amino acids are required by the cultured cells. So they are cysteine, or arginine, glutamine, tyrosine, and non-essential amino acids are also added. And the concentration of amino acids are limited. So it limits the maximum cell concentration attainable, and it balances the cell survival and growth rate. Okay. Then glutamine. So yesterday also have told you the media won't contain glutamine. If you want, we can add the glutamine. So glutamine is required for most cells, and some cell lines will utilize the glutamate. So glutamate is used by cultured cells for the source of energy and carbon, and it is unstable in culture media. And the half-life is only 3 to 5 days. And again, after metabolism, it generates ammonia, which is toxic. Okay, therefore, we have to, if it is necessary only, we should add the glutamate. Glutamax, which is the product commercially available one, is an alanine glutamine dipeptide, which is more stable. Okay, and it is bioavailable due to the action of dipeptidase. So dipeptide, no, so because of action of dipeptidase, it is more stable. Then vitamins. So Eagle's minimal essential media contains only the water soluble vitamins, excluding biotin. So biotin is present in most of the complex media, including serum-free recipes. So biotin is one of the vitamins. Then PABA, para-amino benzoic acid, is present in M199, CMRL 1066, RPMI 1640. Then fat-soluble vitamins A, D, E, K are present in M199, whereas vitamin A is present in LHC9. Vitamin E is present in MCDB 110. So like that, each media is having a particular type of vitamin. And the amino acids vitamin requirements are derived empirically and often related to the cell lines which are originally used in their development. Then salts. So salts are very important for maintaining the membrane potential and many more processes like sodium, potassium, magnesium, calcium, chlorine, sulfate, phosphate, bicarbonates. All these we have to use like to maintain the osmolality. So calcium, it is mainly important for the adhesion of the cells, for the attachment, because catherines are involved in attachment. They require calcium. And it also acts as an intermediary in signal transduction. And the concentration of calcium in the media influence the proliferation and differentiation. And it is reduced in suspension cultures because cells has to get adhered if calcium is there. So calcium should not be used in the suspension culture because it will aggregate. The cells will get attached or aggregate. So to minimize that one, we should not add the calcium, or we can reduce the quantity. Then sodium, potassium, chlorine, they regulate membrane potential. Sulfate, phosphate, bicarbonate, they are required by matrix, and they are nutritional precursors for macromolecules. Then sodium bicarbonate concentration is required for buffering. Okay, sodium bicarbonate and carbon dioxide. Then glucose. Glucose is the most important source of energy. So it is mainly metabolized in the glycolysis to form pyruvate, and it is converted to lactate or acetate. And finally, it enters into the citric acid cycle, and we get carbon dioxide and water. Okay. Then proteins and peptides. So the most commonly used proteins are albumin, protein. So albumin it binds to water, salts, free fatty acids, hormones, vitamins, and they transport them between tissues and cells. And it is a remover of toxic substances from the media. Aprotein is a protective agent in cell culture. So it is stable at both neutral and acidic pH and resistant at high temperature, and also it won't get degraded with proteolytic enzymes. So it inhibits proteases like trypsin. So aprotinin can neutralize the trypsin activity. Then fibronectin. So it is involved in cell attachment. Then transferrin, which will transport the iron. Then fatty acids and lipids. They are mainly important in serum-free media. Then you have trace elements like copper, zinc, selenium, which are required in very small quantities. They are usually present in the serum, but we don't know exactly. Okay, they are required for the proper cell growth. That's why we use serum, because they promote the cell growth. They're called micronutrients, right? Then media supplements, which we can add, are hormones and growth factors. So which we require, if required, we can add. Then antibiotics. So antibiotics are originally introduced in the to prevent the contamination, but there are some disadvantage that if you continuously adding antibiotics, the organisms or the bacteria will become resistant to these antibiotics. And there are cryptic contamination. That means when until you use the antibiotics, there you cannot see any contamination. But there will be bacteria in small quantities and inactive state, and you cannot see, everything will be fine. Once, suppose if any time you are just not adding the antibiotic, immediately you can see the most powerful contamination. Okay, and everything, whatever the bacteria which is there, low level bacteria, that will become more. Okay, and it will also hide a mycoplasma infection. Even the mycoplasma is there, because of adding of antibiotic, you cannot see that. And sometimes these antibiotics will cause anti-metabolic effects also. So these are the antibiotics which we can use: amphotericin B, ampicillin, ciprofloxacin, then erythromycin, gentamicin, kanamycin, penicillin, streptomycin, or tetracycline. All these we can use based on like each and their activity. We can see here. So then the indicator. Okay, this is phenol red. So most of the commercially available culture media use phenol red as a pH indicator, which will allow the monitoring of the pH. The color change, if the pH changes, the color change we can see. At low pH levels, phenol red turns yellow, while at higher pH, it will be purple. Okay, and if it is in the neutral pH, it will be bright red in color. Disadvantages of using phenol red are: it mimics the action of the steroid hormones, particularly estrogen. If you are using anything with the estrogen or studies with the estrogen sensitive cells like mammary tissues, you may get a wrong result. And presence of phenol red in some serum free formulation interfere with sodium potassium homeostasis. And also it interferes with the flow cytometric studies, because in the flow we usually look for the fluorescence, the color change. So that this color will interfere with that color. Okay. Okay, so we will stop here. Okay, and we will continue with the next class. If you have any doubts till here, you can ask. Yes. Do you have any questions or doubts till here? Is it clear?
Yes, ma'am.
Yes, ma'am. Yes, ma'am.
Okay.
Ma'am,
So,
ma'am, this is what you discussed today regarding culture methods and all.
yeah.
The role comes under block 1, fundamental techniques of biotechnology.
Yeah. Animal cell culturing, all these are there, no?
Preparation of culture media.
Culture media, yeah.
That's
yeah, in that. Okay. Yeah. So we will continue the next topics in the next class. Okay, next week. Okay. Thank you so much.
Thank you, ma'am.
Thank you.
Thank you, ma'am.
Thank you, ma'am.